All right, well, here's the lecture for tomorrow, and this one's going to be like our most recent lectures where, we'll have some information in the video. We'll spend a little bit of class time answering any questions that you might have from the video, and then during class on Wednesday, we're going to take it to the next level. This one's going to be a little bit different in that when we take it to the next level, I will show you what uneven crossing over looks like. And also, then we'll go and start to talk about, okay, changes in chromosomal number. Whereas in this lecture, we're really just going to talk about changes in chromosomal structure, except there will be one that can result in changes in chromosomal number. But in our class on Wednesday, we're going to focus a great deal on that. And then on Friday, we will actually take this deeper and go into some current areas of research in this field. So again, just as you prepare for class on Wednesday and you watch this video, just think through any questions that you might have about the mechanism, and we'll see this in class, a big mechanism, especially for duplications. But then also just keep in mind that we're gonna go into a little bit of a different area than what we see in this lecture. All right. So some key terms from this chapter. Cytogenetics. So cytogenetics is the study of the number of chromosomes an organism or a specific cell type has, the structure and characteristics of these chromosomes, and what the genes on the chromosomes actually do, and how the chromosomes can change due to mutations, due to various other chromosomal abnormalities. And so a big thing that's going to come up, and we'll see this time and time again, is when crossing over does not go right. When crossing over does not go right, either it's uneven or it happens between non-homologous chromosomes, it typically results in some chromosomal aberrations. All right, uneven crossing over. So uneven crossing over is typically going to happen on or between homologous chromosomes. It very commonly happens where you have repeated regions, and you get an interesting thing happening. So we talked last time about slippage, where as DNA is being replicated, you might have the two strands separate, and then when they reattach in such a way that you loop out some of those repeats, and you end up copying the template strand over again in a particular region. So that's called slippage. In uneven crossing over, we have something that's called skippage. Basically what happens is you can still form that loop. And again, I'll draw this out for you on Wednesday and hopefully make some more sense of it. But when the a sister chromatid from the paternal chromosome starts to form hydrogen bonds with a sister chromatid from the maternal chromosome, and you get this crossing over event, what can happen is that where they bind, where they start to form hydrogen bonds, forms in such a way that it loops out part of one segment, and you end up basically losing. You get too much of one of those chromatids going with, let's say, you may get too much of the maternal sister chromatid going to the paternal chromosome, and just the right amount of the paternal chromatid coming to the maternal. And so one of your chromatids is going to shorten if too much left, and then the other one's going to lengthen because too much came in. And again, I'll show you how this works, but this, again, like slippage, happens commonly in repeated regions of the chromosome. And then keeping in mind that the centromeres tend to have conserved regions among all the chromosomes, and so this can facilitate non-homologous chromosomes actually getting involved in crossing over. All right, deletions. Deletions happen when you lose a portion of a chromosome. They usually are the result of a single chromosomal break, and so you get a deletion at the end of a chromosome. Under some mechanism, the end of the chromosome might be broken, and so you lose the end of it. And when you just have a fragment of DNA, it doesn't have a centromere attached to it typically, and therefore cannot be bound by a kinetochore microtubule, and therefore cannot be pulled to one of the poles during meiosis or mitosis. And so that piece of DNA ends up usually just hanging out in the cytoplasm. You may get some nuclear membrane that will form around it, and you form like this really small, odd nucleus that doesn't really get inherited properly, and then ultimately it gets destroyed by machinery within the cell. It is also possible to get deletions inside the chromosome, but if that's going to happen, you need a break on either side of that broken piece. Duplications. So duplications happen, we talked about this on Monday, that they can happen due to slippage, where, the mechanism that's replicating the DNA ends up copying the same section twice. Or you can have it due to skippage, which I talked about just a minute ago under our cytogenetics term, where the two chromosomes don't match up perfectly. And so this can happen where you have a lot of repeated regions if you get crossing over between homologous chromosomes. But it happens a lot when non-homologous chromosomes get involved in a crossing over event because they don't match up exactly. And so you end up with these loops forming of sections that didn't match up perfectly. And then the bases that do match up are actually bound. But you end up with too much material coming over in one section, and then DNA machinery writing the complement for that. And we'll see that. We'll see that on Wednesday under these mechanisms. So translocations. Translocations can happen where you basically have one chromosome that gets two breaks on either side of a mobile portion, and then you have a second chromosome that also has two breaks on either side of this, and maybe even a third break to give you multiple pieces of the chromosome. And you usually have this transfer of material because you have this portion of chromosome that gets cut out due to these chromosomal breaks. And then there are machinery. Again, we talked a little bit about this on Monday, or at least in the lecture from Monday, where when you have double-stranded DNA breaks, that there are machines that will match up these breaks and try to match them properly. And sometimes they'll put them into the wrong chromosome. And so usually what you have are what are called reciprocal translocations, in which, say, a portion of, let me give you a very specific example, a portion of chromosome 9 will combine with a portion of chromosome 22, forming what's called the Philadelphia chromosome. And so basically the end of chromosome 9 ends up getting translocated to the portion of chromosome 22, and then that portion of chromosome 22 that got removed got added onto a portion of chromosome 9. And usually these reciprocal translocations are not going to cause a major issue. They're not going to result in a major pathological abnormality, if you will. They will if you move a gene from a regular place in the genome to a more active place, to a place that the chromosome is kept in euchromatin, and the machinery involved in transcription are able to easily access that area. And so when you form the Philadelphia chromosome, what it does is it takes a gene from chromosome 9, which is a proto-oncogene, and moves it over to chromosome 22 to an incredibly active area. And remember, proto-oncogenes are genes that basically a lot of times code for maybe one of those cyclin-dependent kinases, or they code for one of those cyclins. And so when you put this into an incredibly active area, it can lead to overexpression of those genes that contribute to and encourage the cell cycle. And so in this particular example where you make that Philadelphia chromosome, it leads to a specific type of cancer forming called either chronic myeloid or acute lymphocytic leukemia. And we'll talk more about this on Monday. Well, or on Wednesday, sorry. Inversions. So in inversions, typically what will happen is you get a break in the chromosome, and then it's reinserted, but it's reinserted after being rotated 180 degrees. This can happen under a number of mechanisms, many of which happen under kind of a looping event, where the entire DNA chromosome, both strands, form this loop, and then that loop is cut and then reinserted backwards. Now, there are two different types of inversions. There's what are called pericentric, and so these are when the break is actually on both sides of the centromere, and so it actually inverts the centromere as well. And this is typically catastrophic to the functioning of that chromosome. And so if it happens during meiosis, during gamete formation, it can lead to spontaneous abortions. And then pericentric inversions, those are where it happens on the same arm on the chromosome, so it doesn't include the centromere. And oftentimes this can be shouldered. This burden can be shouldered and it not result in any abnormal phenotype. And so it's interesting when you actually compare the genomes of various organisms. Sometimes it'll look like you have an example of an inversion event that happened in the history of that organism. All right, ring chromosomes. Ring chromosomes happen when you have deletion events at both ends of the chromosome. So you get deletion events at both ends. You end up with double-stranded breaks at both ends of the chromosome. And then the machinery that go and try to match up fragments will sometimes match up the two ends of the chromosome and forming a ring structure. And the ring structure it's not going to function correctly. On top of that, you lost both ends of that chromosome, and so whatever genetic material was on the end of that chromosome, you lost. And so these are typically catastrophic events. All right, and so here's a figure out of the textbook, out of this chapter. And it goes through and it shows you basically the way you should look at this is you start down at the bottom here. And so let me grab my pen. You start down at the bottom here and you look at the particular pathology or the particular chromosomal aberration, terminal deletion, an inversion event, an interstitial deletion, ring chromosome, reciprocal translocation, Robertsonian translocation. This is a specific type of translocation that involves the centromere, and I'll show you this, I think, in the next slide, where we've still got a reciprocal translocation, but it involves one of those, at least one of those involved the centromere. And so what often happens is you end up with a chromosome with two centromeres, and you end up with another chromosome that only has one, that has none. And if you don't have a centromere, that chromosome cannot be pulled to either of the poles, and that chromosome can be lost. And I told you at the beginning of this video, we will talk about one thing during this lecture in which you have a change in chromosomal number. The rest of that we'll save for our time together on Wednesday. And so if you look at these particular pathologies or particular chromosomal aberrations, and then go up to the top and look at the number of breaks in the chromosome required to actually create or facilitate that chromosomal aberration. This is how you should look at this graph. And so it's a helpful graph just to know and be able to answer questions of, well, how commonly do some of these things happen, right? The more breaks that they require, the less likely they are to happen, unless there are specific mechanisms to facilitate those types of breaks and those types of chromosomal aberrations. All right, so here's Robertsonian translocation, also called centric fusion, and again, it's a translocation event that involves the centromere. And so here, this fragment that's removed from this chromosome here included the centromere. And so in this particular example, it still generated us 2 chromosomes that each had 1 centromere. But it is possible, say, if this fragment here actually was translocated to this fragment here, rather than the example given, we would create a chromosome that actually has 2 centromeres, and then this fragment would have gone with here, and that would have been a chromosome with 0 centromeres. So a chromosome can't have 2 centromeres, so when it does, through some mechanism, one of those centromeres is deactivated, and then so the chromosome still gets moved properly during meiosis and mitosis. Although you have a chromosome that is not structured correctly, because it's got, 2 sections where you have those very common centromeric repeats. But then here's the real problem. If you get fragment that includes gene F coming over here with the fragment that includes M, N, O, and P, we now have a chromosome that has no centromere. Therefore, you can't get those syn proteins binding properly. You can't get the kinetochore proteins binding properly. If you can't form a kinetochore, the kinetochore microtubules can't bind that, and therefore you cannot pull that to either pole during mitosis or meiosis, and you end up losing a chromosome. It could decrease the overall chromosomal number and lose some genetic information. All right, so that's it that we're going to go over for our class on Wednesday. I know this lecture was a little bit different because we spent a lot of time on that definition page and then didn't have as many slides. But remember, during our time on Wednesday, we'll go over and we'll answer any questions that you have. I want to show you the mechanism of uneven crossing over in both homologous and non-homologous chromosomes. I want to show you a couple other mechanisms, and then we'll go into and talk about what the rest of the chapter actually discusses, and that's how we change chromosomal number other than losing the centromere from one of our chromosomes in a Robertsonian translocation or centromeric fusion.